I have this friend, she's a synthetic chemist,
Hmm.
and I remember visiting her lab a few years ago. She was just sitting in the dark, spending literally weeks staring at these jagged, incomprehensible lines on a computer screen,
Oh, man, yeah.
just trying to figure out if she had successfully built a single molecule.
Right. It's exhausting.
It looked exhausting, yeah.
Yeah.
But that tedious verification process,
Uh-huh.
that's the reality of the physical experimental life sciences, right?
It is. It's a massive bottleneck. We
Oh.
we often think of AI as something that, writes code or generates text.
Right.
But moving that capability into the physical chemistry space, it requires a completely different level of reasoning.
Which really brings us to today's focus on Anthropic's August 2026 research report. We're exploring how AI is fundamentally accelerating these physical life sciences. And before we get into the mechanics of how this works, a quick note. This deep dive is sponsored by Embersilk.
Oh.
Yeah, if you need help with AI training or automation or integration or even software development, or if you're just uncovering where agents could make the most impact for your business or personal life, check out embersilk.com for your AI needs.
Awesome. So looking at the Anthropic report, we're talking about AI directly taking on the most complex parts of drug development,
Right.
starting right at the design phase. Before anyone even runs an experiment, they have to design the molecule.
Which historically has been a painfully slow process, right? Like trying to design a de novo mini binder, it's almost like trying to 3D print a custom Lego brick blindfolded.
Exactly.
And you're hoping it perfectly fits into this tiny disease receptor. So how does an AI handle that computationally?
Well, Anthropic tested Claude, specifically the Mythos Preview and Opus 4.8 models on designing these de novo mini binders. They tested it with 15 different protein targets, and it successfully generated working designs for 14 of them.
Wait, really? 14 out of 15?
Yeah, 14 out of 15. And the hit rates were incredible. Claude achieved hit rates up to 35.1%.
Wow, that's high, right?
Oh, it's huge. The typical industry standard is usually around 10 to 15%.
Oh, okay.
So it's essentially doubling the baseline of what expert human groups achieve.
Because it can explore that biological grammar so much faster. I mean, what stands out to me in the report is the work on TNF-alpha. That's a notoriously difficult inflammatory target.
Oh, yes. It's incredibly complex.
Right, and Opus 4.8 didn't just design a binder for it. It designed a cross-reactive binder that hits the human, monkey, and mouse versions all at once.
Which is historically a massive computational headache. Hitting a multimeric structure across species like that, by getting that cross-reactivity right out of the gate, the AI drastically condenses the timeline for animal studies.
Oh, I see. Because you don't have to redesign it for each phase.
Exactly. It accelerates the entire path to human trials.
Okay, so building a molecule computationally is amazing, but verifying what you actually created in the physical lab, that's where the real bottleneck lies, right? That's what my friend was doing.
Exactly. The physical proof.
So I have to push back a little here. Wait, analyzing raw NMR or LCMS files usually requires proprietary vendor software.
Right.
And like an hour of manual human labor per file. How can a text-based AI do that? If researchers hand Opus 5 an undocumented binary file from a lab, how is it not just hallucinating what the peaks should look like based on its training?
That is the crucial distinction here between a standard chatbot and an agentic model. Opus 5 isn't trying to just read binary data as raw text.
Okay.
When prompted with just a 2-sentence plain text prompt, it leverages a secure code execution environment.
Oh, wow.
Yeah, it writes and iterates its own custom Python scripts to reverse engineer the hex dump. It parses the byte structure, extracts the raw signal data. Basically, it builds its own analytical tool on the fly.
Okay, so it completely bypasses the proprietary software by writing its own parser. That explains how it processed those complex raw lab files in just, 19 to 23 minutes?
Exactly, in under half an hour. And it's doing so with extreme precision. It perfectly matched the human lab's purity assessment at 96.4%.
That is wild.
And the truly remarkable part was watching its scientific judgment in action. During a heavy water check, where they verify the exchangeable hydrogen atoms, the AI initially overstated how much the peaks had shrunk. But because it has that iterative reasoning capability, it reviewed the data it extracted, caught its own overstatement, and corrected its analysis.
No way.
Yes, to match the exact, nuanced conclusion of the expert human operator.
That is so cool. So it's not just running a script. It's evaluating its own outputs critically.
Yeah.
We were talking about taking what used to be hours or weeks of specialized labor, like my friend staring at jagged lines and turning it into a 20-minute autonomous workflow.
And that is profoundly inspiring for the future of medicine. By eliminating these massive operational bottlenecks, AI is fundamentally freeing up human intellect.
Right.
Scientists can step away from the tedious verification loops and focus entirely on discovering life-saving cures. They can move faster and more efficiently than ever before.
It completely changes the scale of human progress. Which really leaves me wondering if AI can now act as both the creative biological designer and the meticulous chemical analyst. What entirely new fields of science will humanity invent with all this freed up time?
Oh, we might be on the verge of disciplines that don't even have names yet.
Yeah, we absolutely are. The tools are finally catching up to our curiosity. Well, if you enjoyed this podcast, please subscribe to the show. Hey, leave us a 5-star review if you can. It really does help get the word out. Thanks for tuning in.